The only fatty acid that our bodies can synthesize is palmitic acid, but we are able to oxidize any of the different fatty acids that we ingest. My name is Alyssa, and in this video we will investigate the physical and chemical mechanisms of fatty acid metabolism. We'll start out by examining the nomenclature of fatty acids. Then, we'll move on to investigate the synthesis and oxidation of fatty acids within our bodies. After watching this video, you will be familiar with the synthesis and oxidation of fatty acids, and you will be comfortable answering questions on this topic as you study for test day. First, we will analyze how we name fatty acids based on their structures. We'll start by analyzing a saturated fatty acid called palmitic acid. This is the only fatty acid that our body is able to synthesize. In the name, we see the numbers 16 and 0. The first number relates to how many carbon atoms are in the fatty acid chain, starting at the carbonyl carbon and counting all the way down to the end of the chain. When we talk about the specific carbon atoms in the chain, they are labeled using Greek letters so that we can easily identify them. The carbonyl carbon is not labeled as it is easily distinguishable, and so we begin labeling with carbon 2, or the carbon just beside the carbonyl group. This is given the designation alpha, and the adjacent carbon is beta. The second number in the name relates to the number of double bonds present in the fatty acid. For palmitic acid, there are no double bonds, and therefore the fatty acid is said to be saturated. Unsaturated fatty acids, like linoleic acid, have one or more double bonds in the fatty acid carbon chain. Like before, when naming, the first number depicts the number of carbon atoms in the chain, and the second number, in this case a 2, depicts the number of double bonds. We must also identify the geometry of these double bonds when naming fatty acids. In linoleic acid, we can see that both the double bonds are in the cis, or same-side conformation, and this is indicated by writing cis twice within the name, because both double bonds have this geometry. This is the most common geometry for fatty acid double bonds. It is then important to state where these double bonds are found. Starting from the carbonyl carbon, carbon 1, we count the carbon atoms in the chain until we reach a double bond. In this case, the first carbons of each double bond come at carbons 9 and 12. There is another system of naming these unsaturated fatty acids called the omega numbering system, where the omega carbon is the last carbon in the chain. In this system, the omega tells us the position of the last double bond relative to the end of the carbon chain. The last double bond in linoleic acid is 6 carbon atoms away from the end, and so this fatty acid is a member of the omega-6 family. Before we introduce the synthesis of fatty acids, we need to explore what happens in our bodies so that we get a buildup of acetyl-CoA, a molecule used in the synthesis of fatty acids. Typically, acetyl-CoA is used in the mitochondria in the citric acid cycle and produces NADH, FADH2, and GTP. From there, NADH and FADH2 are used in the electron transport chain, and molecules of ATP are made. When the energy needs of the cell are met and ATP is high, the electron transport chain is inhibited. This reduces the amount of NADH used, leading to a buildup of it. High levels of NADH and ATP inhibit the citric acid cycle, which of course will then limit the use of acetyl-CoA and cause a buildup of it in the mitochondria. When there is a buildup of acetyl-CoA, it is used in the synthesis of palmitate. This synthesis occurs in the cytoplasm and requires 8 molecules of acetyl-CoA. But we know that the buildup of acetyl-CoA is in the mitochondria, and unfortunately, the mitochondrial membrane is impermeable to acetyl-CoA. This means that acetyl-CoA must be converted to something that can pass through the membrane. This is accomplished through the addition of oxaloacetate to form the membrane-permeable molecule citrate. Once in the cytoplasm, acetyl-CoA can be reformed from citrate using ATP. We now have acetyl-CoA in the cytoplasm for fatty acid synthesis. But what about the molecule of oxaloacetate? This molecule can be converted to malate, and from malate to pyruvate, producing NADPH via malic enzyme. Pyruvate can then enter the mitochondria and be converted to oxaloacetate by pyruvate carboxylase or to acetyl-CoA by pyruvate dehydrogenase. The acetyl-CoA can then be converted to citrate and sent to the cytoplasm for more fatty acid synthesis. Now let's explore the synthesis of palmitate from acetyl-CoA in the cytoplasm. First, acetyl-CoA is carboxylated and converted to malonyl-CoA via an endergonic reaction. The enzyme at work here is acetyl-CoA carboxylase, and this enzyme requires the cofactor biotin and a molecule of ATP to function. This is the rate-limiting enzyme within the synthesis of fatty acids. Malonyl-CoA is then reduced and decarboxylated and can be incorporated into the growing fatty acid chain. The enzyme at this step is called fatty acid synthase and requires a molecule of NADPH to reduce the acetyl group of malonyl-CoA. As mentioned before, fatty acid synthesis occurs when energy is abundant. Insulin, whose levels are high when energy is abundant, stimulates acetyl-CoA carboxylase and fatty acid synthase to promote palmitate synthesis. Let's take a closer look at the molecules and mechanism associated with this synthesis. How does a 2-carbon molecule become a 16-carbon molecule? The first step is the conversion of acetyl-CoA to malonyl-CoA through carboxylation. Here we can see a little bit easier how malonyl-CoA is just the addition of CO2 to acetyl-CoA. At the same time, a molecule of acetyl-CoA is being bound covalently to a part of the fatty acid synthase enzyme called the acyl carrier protein, or ACP. The malonyl group is also activated in the same manner before the reaction proceeds. Then, 2 molecules of NADPH are used to reduce the acetyl group while the malonyl group is decarboxylated. In this process, these 2 molecules are combined. Take note that the acetyl from the malonyl, indicated in red on your screen, is beside the acyl carrier protein in the final molecule. To continue forming the fatty acid chain, malonyl-CoA continues to be formed from acetyl-CoA. It is then decarboxylated and added to the growing fatty acid chain. Note once more that the newly added acetyl group is still beside the ACP molecule, and that the new acetyl groups are always added in this position. This process continues until the entire fatty acid chain has been synthesized. Now, the synthesis of the 16-carbon fatty acid palmitate is complete. Altogether, it takes 14 molecules of NADPH to complete this process. It is important to note here that acetyl-CoA carboxylase performs the conversion of acetyl-CoA to malonyl-CoA, while ACP linkage and the following steps are performed by fatty acid synthase. Once synthesized, the fatty acids are stored as triacylglycerols until energy is required later. They then undergo oxidation in the mitochondrial matrix to release molecules of acetyl-CoA that can enter the citric acid cycle for energy. Fatty acids taken up from the blood must be transported to the mitochondria from the cytoplasm. Small to medium chain fatty acids of 1 to 12 carbons can pass easily into the mitochondrial matrix, but larger fatty acids of 14 to 20 carbons require assistance to get into the matrix. In the intermembrane space, the fatty acid is activated by linkage to coenzyme A. This step facilitates binding by carnitine, which is necessary for transport into the mitochondrial matrix. A special carnitine transporter is then used to do this. Once in the matrix, the fatty acid is again linked to CoA, making a fatty acyl-CoA molecule, and through the process of beta oxidation, acetyl-CoA will be produced. This process depends on the concentration of fatty acids within the blood, and will not move forward if these concentrations are low. But malonyl-CoA can inhibit the carnitine transporter, which would also prevent beta oxidation from occurring. Let's examine the mechanism of oxidation for a long fatty acyl-CoA molecule, leading to the release of acetyl-CoA. Notice that there is an acetyl-CoA equivalent in the first 2 carbon atoms. Beta oxidation gets its name because this series of reactions will oxidize the beta carbon, thereby releasing a molecule of acetyl-CoA. The first step forms a double bond between the alpha and beta carbons. Here, we have simplified the fatty acid chain so that the screen doesn't get too cluttered. Then, water is added across the bond to add a hydroxyl group to the beta carbon. NAD+ is reduced to NADH as the beta carbon is oxidized to a carbonyl group. And finally, the beta carbon forms a bond with an additional CoA molecule, which splits the fatty acid, yielding 2 CoA molecules. The first 2 carbons have been separated into acetyl-CoA, and we are then left with a fatty acyl-CoA molecule that can undergo beta oxidation. And so beta oxidation does occur again, and this time the next 2 carbon atoms are involved, so our shorthand fatty acid chain decreases from C12 to C10. The new beta carbon is then oxidized through the same process as before, and the cycle continues until the entire fatty acid chain has been broken down into acetyl-CoA. In this process, however, there is a difference in the last step when oxidizing odd or even-numbered fatty acid chains. Before we examine the last step in the oxidation of odd-numbered fatty acids, let's finish the process with even-numbered fatty acid chains first, since these are the most commonly oxidized fatty acids. On the last step, even-numbered chains will oxidize to 2 molecules of acetyl-CoA. These molecules are then used within the citric acid cycle. Remember that 1 molecule of acetyl-CoA takes 2 carbon atoms from the fatty acid chain. Odd-numbered chains are slightly different. After oxidation, 1 molecule of acetyl-CoA will be made from the 2 carbons adjacent to the CoA and enter the citric acid cycle. Now we are left with a 3-carbon chain. These carbons then undergo a series of enzyme-catalyzed reactions to eventually form a succinyl-CoA molecule. Succinyl-CoA doesn't directly enter the citric acid cycle, but must first be converted to malate. Malate can then be transported to the cytoplasm and converted to pyruvate by malic enzyme. Pyruvate can then enter the citric acid cycle in the mitochondria or participate in gluconeogenesis. This is an important detail here. Odd-numbered chains, in contrast to even-numbered chains, can be converted to glucose. Unsaturated fatty acids must undergo additional reactions to continue beta oxidation because with the presence of unsaturation, the enzymes required for oxidation do not recognize these substrates. Consider linoleic acid. The first 6 carbons can be oxidized through beta oxidation, but after 3 rounds, a double bond is present that sterically hinders the enzymes involved in oxidation. Enoyl-CoA isomerase will isomerize the double bond to an alpha-beta double bond, which is found 1 carbon away from the carbonyl group. This molecule can then complete 1 round of beta oxidation, starting at the second step of the process, so when water adds across the double bond, and 2 carbon atoms are removed from the chain. Then, another round of oxidation can begin, but stops after only the first step forming the double bond. That's because of the conjugated molecule that's formed. This molecule no longer fits properly into the enzymes needed for the oxidation, and so another enzyme must be used in order to make it a suitable substrate. Now, the double bonds are found at carbon 2 and carbon 4, and the enzyme needed is 2,4-dienoyl-CoA reductase, and uses NADPH to reduce one of the double bonds, resulting in a carbon 3, carbon 4 double bond. An isomerase then converts this to a carbon 2, carbon 3 double bond. This substrate is now familiar, and reacts through beta oxidation starting at the second step again to break down the fatty acid chain and produce molecules of acetyl-CoA. Now that we are familiar with naming, synthesizing, and oxidizing fatty acids, let's test what we have learned with a few practice questions. This first question is testing your knowledge of naming fatty acids. The answer is B. Answer choice B has 20 carbon atoms, 5 double bonds, and the last double bond is 3 carbons away from the end. This corresponds to all of the features present in the name of the fatty acid given in the question stem. You can use the number of carbon atoms to eliminate the other answer choices. This second question is asking you to think about why beta oxidation is inhibited after a meal, despite high fatty acid concentration in the blood. The answer is B. Low beta oxidation is due to the stimulation of the enzyme acetyl-CoA carboxylase by insulin. Acetyl-CoA carboxylase produces malonyl-CoA, which inhibits the carnitine transporter, thus reducing fatty acid transport into the mitochondrial matrix where beta oxidation occurs. Answer choice A is a true statement, but this does not directly affect beta oxidation. This enzyme is used in fatty acid synthesis. Answer choice C is false, because insulin actually promotes fatty acid uptake by the liver to make triacylglycerols. Finally, answer choice D is a true statement and would lead to higher fatty acid concentration in the blood. However, after a meal, glucagon levels should be low and would not contribute very much to the concentration of fatty acids in the blood. This final question is testing your knowledge of beta oxidation as well as your skills in naming fatty acids. The answer is A. 9 acetyl-CoA molecules can be made from 18 carbon atoms. Only 8 turns of the cycle are required because for even-numbered fatty acids, the last step generates 2 molecules of acetyl-CoA. Before we finish, let's quickly recap the important points surrounding fatty acids. In regards to naming, the number of both the carbon atoms and the double bonds must be included in the name. The synthesis of palmitic acid occurs in the cytoplasm from acetyl-CoA. The first step is rate limiting and requires the enzyme acetyl-CoA carboxylase. This forms malonyl-CoA, which forms palmitic acid in the presence of fatty acid synthase. Remember that both of these enzymes are stimulated by insulin. Fatty acids are broken down by beta oxidation. This reaction occurs in the matrix of the mitochondria, and in order for large-chain fatty acids to get into the matrix, they must be transported by the carnitine transporter, which is inhibited by malonyl-CoA. In beta oxidation, 2 carbon atoms are removed at a time. For even-numbered fatty acids, 1 acetyl-CoA molecule is made for every 2 carbon atoms in the chain, and for odd-numbered fatty acids, the last 3 carbon atoms of the chain are used to synthesize pyruvate. Finally, we saw that polyunsaturated fatty acid chains required special enzymes to isomerize the double bonds into substrates that can be recognized by enzymes used in oxidation. Thanks for watching this video. Good luck studying for your MCAT.